but there's not a failure of activating an appropriate response to control and contain most of the parasites in this individual. And that's certainly not true in these self-resolving cutaneous lesions. It just takes a long time. So there is naturally acquired resistance. There are no real vaccines. But let's talk first about the nature of the acquired immunity that might occur in these individuals and that we can study in these really nice mouse models. So the favored model of leishmaniasis in an animal is the laboratory mouse, and that's because I'm sorry. The favored model of Leishmania strains to use is Leishmania major, and that's because it normally infects rodents. So the laboratory mouse is a fantastic model to in which to study the nature of acquired resistance to Leishmania infection in mouse models in the lab. So again, this is the geographic distribution of Leishmania major. The lesions are typically localized to extremity sites, like on the hand, 1 or 2 lesions, and they will heal in the absence of treatment, but they can occasionally be pretty severe. Here's an Afghan child with multiple lesions on the face. These are the natural sandfly vectors. In our vector biology discussions, we talk mainly about Papatazi, which transmits in these areas. Very closely related fly, Phlebotomus dubowski, transmits in this region of Africa. And remember, we were talking about the interesting parasite-vector interactions which produce these species-restricted vector capacities for Papatazi. Papatazi and dubowski will only transmit Leishmania major parasites. So we have these flies in the lab, and the data I'm going to show you we're going to look at immune responses in mice that are transmitted by Phlebotomus dubowski. So a lot of labs that study L major infection in mouse models, they use the C57 black 6 mice because it's the classic genotype that is resistant to L major infection. A lot of people put parasites in the foot pad. Our preferred site is to put it in the skin because that's where a sandfly would normally deposit these parasites. So when we and other groups use needle inocula, we put it in the skin of a mouse, and they'll develop these nodules. Sometimes they'll ulcerate, usually not, but they'll always self-heal within a few weeks or a few months. And these pathologies look quite a bit like the kinds of lesions you see in people. So that's the model we use. And that's the model we and a lot of people have used to, the black 6 mouse is a fantastic laboratory mouse because there are so many knockout lines, immune deficient lines, that have been knocked out for various immune related molecules involved in Th1 immunity. And so over the years, this mouse has very clearly demonstrated the mechanisms underlying acquired immunity during these infections. So if you put L major again, these are needle challenges. In the footpad or the ear, in INOS knockout mice, IL-12 knockout mice, either the P35 or P40 chain, CD40 ligand knockout mice, and of course interferon gamma knockouts. Or if you treat the mice with antibodies against anti-CD4, anti-CD8, or CD8 knockout animals, they'll all develop uncontrolled lesions and uncontrolled parasite loads in the skin. So the dotted line is the parasite numbers in the wild-type mice. So these parasites grow beautifully for a few weeks, but then they takes a long time, but you start to kill them pretty efficiently. So you'll kill off 99% of these parasites in the skin, and they'll develop these nodules that aren't very severe, and then they'll heal. So in the knockout lines, so here's an example interferon gamma knockout mouse. They develop these really severe pathologies. Non-healing lesions start eroding the ear-dermis, destroying the tissue, and huge numbers of parasites are left uncontrolled in the skin. So acquired resistance in this model involves induction of a strong Th1 response. It's IL-12 driven. That's the instructive cytokine from innate cells that differentiate the CD4 precursor cells to differentiate to Th1s. They signal along a CD40 ligand dependent activation pathway. That's a critical pathway for IL-12 responses, as we discussed a bit yesterday. And these, this leads to activation of both CD4 and CD8 positive T cells to secrete high levels of gamma. So both CD4s and CD8s contribute to this acquired resistance. Okay. So there were literally hundreds of papers looking at immune-deprived, genetically deficient mouse in this model to demonstrate. It was one of the earliest and best described in vivo infection models to show how important Th1 cells were, the cytokines and the factors involved in Th1 differentiation, to show how important these pathways were to an intraphagosomal parasite like Leishmania. So it has a lot of relevance to other intraphagosomal parasites like tuberculosis, right? Simplistically, these are the cells and the cytokines involved in killing of these parasites. So dendritic cells will take up antigen or parasites and present parasite antigens to a T cell, and in conjunction with upregulated CD40 and CD40 ligand interactions and CD80, CD28 interactions, plus IL-12. So probably 3 signals are required to get strong Th1 development through the T cell receptor, recognizing antigen, these co-stimulatory molecules, and IL-12 cytokine. And then these cells will make a lot of interferon gamma, and they'll act on these infected macrophages or monocytes, whatever phagocytes have taken up these parasites, to upregulate inducible NO-synthase, and you'll get production of a lot of nitric oxide, which is diffusible, and start killing these parasites. So that's how that works. There's no reason to think that these same mechanisms don't also work in humans the same way. Okay. another look at this mouse model, this Block6 resistant mouse model. And we sort of divided into sort of 3 phases of infection. The initial phase, which can last some weeks, is parasite growth and the T-cell priming phase. So in black is parasite growth, in red is lesion development. You start killing these parasites after a few weeks, and you contain their growth and you kill most of them. But to emphasize again, there is never a sterile cure. There's always some parasites left. So, and we're convinced that's true in people. We discussed, I guess it was yesterday, why that might be, in terms of these tissue-resident macrophages that, even in the presence of high levels of interferon gamma, are not activated for killing. So they're a beautiful intracellular niche for these kinds of pathogens. And it's how they can establish a persistent infection even after healing. So there's really complete resolution of these lesions, and yet a persistence of some hundreds or thousands of parasites in the site, and there's parasites in local draining nodes as well. Okay, so that's the parasite maintenance, this chronic phase. What's important here is that these healed primary infections vaccinate or confer strong immunity against secondary infection. So despite the fact there's not sterile cure, if you re-challenge, say we'll re-challenge the other ear. This is the secondary challenge on another site. We'll use the other ear of the same mouse. They don't develop any lesions at all. So they're beautifully immune. And you can wait weeks or months. In these mice that have healed their primary lesion, you can wait for the whole life of the mouse and challenge, and they'll still be immune. So this is a beautiful model of acquired resistance. And we would love to know how this works. So many labs have studied the nature of this immunity, particularly because it's the basis of the same kind of acquired resistance you see in people, and the basis for a procedure called Leishmanization that has been used for generations to protect people in these endemic regions against cutaneous leishmaniasis. And what this involves is the deliberate inoculation of virulent Leishmania major on selected sites, usually the arms, sometimes the buttocks. And when those lesions, those deliberately inoculated lesions heal, these individuals have strong protection against a natural exposure. So I want you to appreciate, this is not really a vaccine. This is, they're being given leishmaniasis. But it's a controlled dose. The lesions are typically not so severe, but they are chronic. They take a while to heal. And it's not an attenuated strain. So it's not really a vaccine per se. But you know, you could argue it's like a vaccine. It's not delivered by flies. It is cultured parasites that they're inoculating. So they may, in fact, be attenuated compared to what flies actually deliver. And you're using a selected site. That's critical. But whatever you want to call it, a vaccine or not, this is the only procedure that uniformly protects humans against any form of parasitic infection. So there are no vaccines against malaria or sleeping sickness or any helminth infections. So this is the one the one experience that that, whether you call it vaccination or not, confers just solid lifelong immunity in these individuals. There were, again, this Leishmanization was started long time ago, so I think it probably started in the former Soviet Union. Here's a controlled trial in 1940. They took 1,000 subjects and Leishmanized, needle injected these cultured parasites in 1,000 of them, and 1,000 were controls. And this was a highly endemic area for Leishmania major transmission. After a natural exposure, I think the follow-up was 1 or 2 years. 6% of the Leishmanized group developed cutaneous leishmaniasis, 64% of the control group. So a lot of transmission going on in this area, but fantastic protection. Israel took up this same this same Leishmanization protocol in their populations in the 70s, and they did a controlled trial. Here there was complete protection, 0% lesions in the Leishmanized group, 30% in the control group. And in Iran, that has been the country that has been doing Leishmanization in most recent times, although I think they stopped doing it in the late 80s or the 90s for reasons we'll discuss. Again, just 0.8% incidence of leishmaniasis in the Leishmanized group. Sometimes they needle inoculated these parasites and they were non-takes, and they even still saw some protection. And compared to the controls, again, a high transmission area, 40%. So again, I want to stress that if you consider this a vaccine, this is the only vaccine against any human parasitic infection that actually works, and will work almost every time. So why not just use this? It's at least against cutaneous leishmaniasis. This was homologous protection. They're using L major to protect against L major natural exposures. So these are the sort of problems that preclude its widespread use. First of all, these are chronic lesions. They last a long time. And even in these endemic settings, these families are reluctant to have their children leishmanized because they'll constantly pick at these lesions and they can develop secondary infections. So it's not something you're anxious to give to children, although you're anxious to immunize children. It's believed that there's not sterile cure, so the parasites persist, and so there is a risk of reactivation. Although with Leishmania major infection, there is very little reactivation that has ever been described. It's a live inoculum, difficult to standardize, and there's a lack of quality control, and just the delivery of live inoculum to these populations. So the practice is occasionally taken up in some endemic. I think the last place that I know of was Uzbekistan. They had a terrible problem with cutaneous leishmaniasis in new settlement areas. So when new settlements in these agricultural regions, when human populations suddenly are living in close proximity to these rodent reservoirs of infection, there's just an explosion of cutaneous disease. So I think in the late 1990s or even later, they took up Leishmanization to protect these populations. But other than that, this practice has been discontinued. This just gives these are a couple of Iranian individuals that were Leishmanized, and these are just, there's one individual on top, another on the bottom, and it just shows the nature of these cutaneous pathologies and how long they last. So, you know, at least half a year in each case, there's still some lesions. Because they have cutaneous leishmaniasis. It's with a virulent strain of L major. Okay, so the fact that there was this strong naturally acquired immunity to a single primary infection, that's the kind of experience that in the past non-living safe vaccines have been developed and been shown to work. So zoonotic cutaneous leishmaniasis, this experience of natural immunity acquired after a single infection, it was believed that, well, this should be one parasitic infection or one form of leishmaniasis that we should develop a safe vaccine against. And these are the reasons for optimism regarding the development of safe and effective vaccine against at least cutaneous leishmaniasis. The protective immune responses are induced following infection and recovery in the majority of forms of at least cutaneous leishmaniasis. And several protective antigens have been identified, and they're highly conserved among parasite isolates and species. There's not a lot of strain variability that prevents, you know, vaccines against things like malaria or African trypanosomes. The mechanism of protection is that we, you know, reasonably well understood from animal models, and the clinical studies support most of those findings. Recently developed adjuvants capable of promoting the right sort of T-cell response, these Th1 responses, are available. And again, these really good animal models for testing vaccines. So this is just a partial list of all the preclinical studies on various vaccines that have been developed against mainly cutaneous leishmaniasis. And almost all these studies, a couple were done in dogs, but most of them in mice. These are both DNA-based and protein-based subunit vaccines, recombinant vaccines, against certain target antigens like GP63, that's the metalloprotease we discussed before, and various other Leishmania proteins, histones, some other cell surface proteases. So lots of different target antigens, which when delivered with the right sort of Th1 adjuvant, in some cases BCG, in some cases recombinant IL-12 was used, in some cases CpG. So these are the various adjuvants, and this shows the protection. It was never complete protection, but always pretty good, you know, pretty good protection against these parasites loads at the site of challenge. So lots of potential vaccines. There's just lots of them out there. And the easiest one that a lot of people have used is we can grow the insect stages of the parasite, the pro-mastigoid forms, cheaply in culture, and you just kill them. You autoclave them, so you heat kill them. So it's autoclaved Leishmania major. And you give it with a Th1 adjuvants like CPGs or IL-12, and it provides strong protection against needle challenge in the mouse. And so keep in mind, all of these studies were evaluated using needle challenge, not natural sandfly challenge. So based on a lot of these studies, a lot of human trials were done. And this is a list of Leishmania vaccines that have been employed in human subjects. The ones up here are the relevant ones in terms of these were actual field efficacy trials, phase 3 efficacy trials of vaccines. So there were a couple of field trials. Where was this one done? I don't know where that. No, this was done in Brazil. These were done in Brazil. These were heat killed. No, these are Iranian studies. These were done in Brazil. These were just whole cell heat killed vaccines with or without BCG. Here's recombinant. This was phase 2 trials, recombinant. The point is there was really no protection. They were found to be safe, fairly immunogenic, but no protection. This is a meta-analysis of the phase 3 trials of killed whole parasite vaccines. So again, just these autoclaved whole cell pro-mastigote vaccines with BCG, live BCG, because in many of these regions, children get BCG vaccination to protect against tuberculosis. It's a poor vaccine against TB, but there's a huge experience with it. It's thought to be safe, and it's thought to promote, at least in part, a Th1 response. So it was used as an adjuvant. So these are the relative risks after 1 or 2 year follow-up of these vaccines. And these are different sites in Iran. This is Sudan. This is Brazil, Colombia, Ecuador. So there was never any reduction in the relative risk except this one trial in Ecuador, which could not be repeated. Otherwise, there was just no difference in the people who received this vaccine as compared to controls, which were BCG alone. So it just didn't work. So why is that? Why do these things work pretty well in these mouse models, but not in humans? Well, there's a lot of differences between mice and humans, but just in terms of how these trials were done, how the vaccines were evaluated, the mice were evaluated with a needle inoculum. And these trials, obviously, this is a natural exposure. These vaccines had never been actually tested against a real sandfly challenge. It was just putting parasites in a syringe and sticking them in a mouse. So that was a possible difference. Seemed like it could be pretty important. And because we had flies, it was something that we could be attentive to. So we had the flies, and so let's take these vaccines, these killed vaccines that have been shown to work pretty well in a mouse, and compare a proper sandfly challenge with a needle challenge. So that's what we set these experiments up to do. We can just consider the possible differences between needle and sandfly inocula. We've already talked a bit about it. But an important one is the dose and infectivity of metacyclic promasticates. You can't control that in flies. With needle, you make a somewhat, folks have made a pretty arbitrary decision what dose of parasites to put in. And in terms of infectivity, a lot of these challenges were not done with with real metacyclics. And we now know sandflies delivered just these beautiful metacyclics in variable doses, but a lot of the needle challenges were not well-defined inocula with regard to had they differentiated to metacyclics or not. There's tissue damage associated with fly probing and biting behavior. So we looked at that the other day. That really stressed the impressive amount of tissue damage that results in this massive inflammatory reaction. And neutrophils swarming in these bite sites, and we don't see that that kind of very localized inflammatory reaction to a needle injection. And then, of course, flies are not just delivering parasites. They do salivate. They deliver components in the saliva that have inflammatory and immune modulatory properties. And we also discussed that they may be delivering the microbiota from their guts as well. So there's a lot of these possible differences between injecting just parasites by a needle and a fly, and a natural transmission by a fly bite. This just reminds you of this massive neutrophil infiltrate at the site of a bite within the first hour or so. Right? So this is what a challenge transmission site would, this was done in a naive mouse, but if we now immunize a mouse and challenge it with an infected fly, it's going to look the same. So just to remind you, this is our infected fly challenge model. We take, we seed mouse blood with tissue A masticates. We infect the flies and wait for a couple of weeks so we get these beautiful anterior infections with metacyclic parasites in the flies. And then we put the flies in a vial and expose the mouse ear to the bites of a few of these infected flies. So we're comparing a live vaccinated mouse. Just to be clear, when we say live vaccinated, we mean a Leishmanized mouse, a mouse that has had a primary lesion that healed. So this mouse would have had a primary infection with L major in the foot pad, and we let it heal over 2 or 3 months, and now we're re-challenging it with either needle or fly bites a few months later. Okay, I guess 18 weeks later. And we're comparing these live vaccinated mice with a healed primary lesion to mice that have been immunized with this killed vaccine. This is the killed vaccine that was used in many of the human vaccine trials in Iran, this autoclaved Leishmania major plus CPG. And then we're going to challenge either by needle or infected fly challenge these weeks later and analyze the tissue. So this is 4 weeks after needle inoculation or infected sandfly bite. This is the control. This is the killed vaccine. By needle challenge, you see each of these is 1 ear of a mouse. The total parasite loads in the ear of the challenge mouse. Again, these are needle challenges, so we can beautifully control the inoculum size. So all the mice are behaving pretty similarly. And there's a 1 to 2 log-fold reduction, so that's pretty good immunity. It's not great, but it's not bad. When we do infected fly challenge, again, the first thing you'll notice is the range of parasite loads when you use flies to transmit infection that delivers such a range of doses. So we have to use a lot more mice. But what is obvious here is that this killed vaccine didn't confer any protection against fly challenge. So there is a difference. But importantly, if you take the healed mice that have had a primary infection that healed, it confers much stronger protection. Now there's just between 10 and 100 parasites in that challenge site. So it's really strong protection conferred by the killed vaccine. And more importantly, the healed mice have just fantastic immunity even against infected sandfly challenge. So a healed primary infection, even against needle challenge, is still the sort of gold standard of immunity. And vaccines should always be recombinant or subunit vaccines should always be measured in parallel studies with the strength of the protection conferred by the gold standard of immunity, which is a healed primary infection. Because that's the only thing that confers immunity against a natural fly challenge. So if this immunity by a killed vaccine against needle challenge is not as strong as seen here, it's probably not going to work against fly challenge. Okay. So you don't. So the argument is you don't necessarily need flies to evaluate your vaccine, because hardly anyone has flies. These are hard experiments to do. But you should compare the efficacy of your vaccine against the strength of the immunity seen in a mouse with a healed primary infection, because that's the gold standard. So that's one argument to be made. So here's another vaccine that's in clinical development. It's a recombinant subunit vaccine, a trifusion protein. These are 3 different Leishmania antigens, TSA, LMST11, and LEIF. It's being made by the group out in Seattle, Steve Reed's group. And they're making it both as a therapeutic vaccine and as a prophylactic vaccine. And this is the adjuvant they use, monophosphoryl A in a stable emulsion. It's a pretty good adjuvant being used in the malaria vaccine trials. So same deal. This vaccine, this is formulations of their vaccine with this adjuvant. It confers some protection compared to the age-matched controls against needle challenge. No immunity. Again, each of these circles is the parasite load in the ear of a single mouse. And I think it's 4 weeks post-challenge. And this formulation of the recombinant vaccine actually made things worse. Okay. But healed mice conferred the best immunity, again, against needle challenge, and importantly, healed mice also protected mice against natural sandfly challenge. So we were interested in studies published a long time ago. So is this acute neutrophilic response, does it play any role in compromising the efficacy of these vaccines against sandfly challenge? So neutrophils were depleted just prior to the infected sandfly challenge. Again, so all of these mice, this is sandfly challenge, and that's noticeable immediately because when we evaluate these mice at 5 or 6 weeks post-infection, there's a huge range in parasite loads in the challenge site that you don't see when you needle challenge because you control the dose. But against fly challenge, still the only significant protection was the mice that were healed, had a healed primary infection. When we neutrophil depleted the mice, the ALM vaccine did confer some significant protection compared to the age-matched controls. So it did rescue the efficacy of this vaccine, at least partially. So all that tissue damage, all these that acute neutrophilic infiltrate did compromise, and we discussed how that response promotes the infectious process so beautifully. Picks up these parasites, delivers them to dendritic cells and to dermal macrophages, and prevents innate responses and adaptive responses, or delays the onset. So, but depleting neutrophils at the time of challenge is there's no practical way to do that, to rescue a vaccine, because you would have no idea. The neutrophil depletion lasts only a few days, so you would have no idea when individuals were going to actually be bitten by an infected fly. So this was just a proof of principle that infected fly bites do provoke the kinds of reactions that make immunity difficult to be expressed. So let's talk about the kinds of cells in these healed mice. So what's the vaccine not doing that we would like them to be doing, and to confer immunity, and that mice with a healed primary infection, why are they so powerfully immune to even a sandfly transmitted infection? So we'll just consider for a bit the nature of the CD4 cells that are activated and that confer the different kinds of immunity. And I'm sure you've heard these terms. There's effector cells and central memory cells and effector memory cells. We're not going to talk much about these. These are really discussed more in effector memories in the context of viral infections and CD8 responses. So let's really focus on the difference between effector cells, which are the cells that actually do the work, that go to the sites of infection and start releasing the cytokines to activate cells for killing. So the effector cells have these markers. They're Ly6C positive. CD44, when they're CD44 high, it means they're an antigen-experienced cell. That distinguishes them from a naive T lymphocyte. So both central memory and effector cells are antigen-experienced. They both are CD44 high. So that's a beautiful marker to just distinguish them from naive cells that are coming out of the thymus and have not yet encountered antigen at all. What central memory cells do is they maintain expression of CD62 ligand, which is a selectin, which allows them to home into the lymph nodes. And central memory cells position themselves in these lymphoid tissues, waiting to be reactivated by dendritic cells which have carried antigen from the periphery to the lymphoid tissue. So CD62L is this important selectin to get these memory cells in the right place to be reactivated for a secondary response. Effector cells, by contrast, downregulate this so that they're permitted to recirculate in the blood, in the periphery. And they're tissue homing. They're looking for sites of inflammation, some insult in the skin or wherever, in the lungs, and then they'll penetrate that tissue, they'll home to that tissue, and they'll immediately release cytokine. So they don't divide, they're just recirculating. But the problem is they're pretty short-lived, and they require constant renewal by antigen. But they're activated, antigen-experienced, tissue-homing cells which confer at the site of insult, at the site of a secondary challenge, very rapidly the ability to activate cells for killing. These cells will do the same. They will give rise, but they will give rise to effectors, but they take a while. They have to receive antigen from the periphery to the draining nodes. They have to undergo proliferation. They have to differentiate into cytokine secreting cells, and then they have to recirculate, retraffic back to the periphery, to these peripheral sites of challenge. So it takes much longer. So these can be maintained by ongoing infection. These don't require antigen. These are much more long-lived even in the absence of antigen. So a vaccine, a non-living vaccine, and that has been the paradigm for vaccine development all along, is this is the cell you need. You just need a central memory cell and non-living antigen. Conventional antigen can generate those cells, and that should be sufficient. So to which of these subsets, CD4 subsets, T-cell subsets, are associated with the protection we've been talking about in these mice? So what we'll see is that early Th1 immunity at the dermal site of challenge by infected fly bites is the best correlate for protection. So we're going to compare in the skin, in the challenge site, and we're comparing we lost the label of the days here. I think this is 3 days post-challenge. Do you see that anywhere? No, I don't see it. This is 3 days post-challenge. This is 7 days post-challenge. So that's critical. This is really a short time after challenge. In the ears of mice, the CD4 cells secreting gamma that come into the challenge sites at 3 days and at 7 days in mice receiving killed vaccine and in mice that had these healed primary infections. So you don't see anything at 3 days in the vaccinated mice in the challenge site. And again, these are sandfly challenge sites. So, and you begin to see a few cells, CD4 cells secreting gamma at 7 days. But that's pretty delayed immunity. And again, these killed vaccine mice were not protected at all against fly challenge. Whereas these healed mice, these mice that have healed a primary lesion, already by 3 days, you see pretty good numbers of CD4 cells secreting gamma in the challenge site, and huge numbers already by 1 week. I mean, so this is a fantastic response. So it makes sense. These mice are so well protected. You're just getting lots of cells secreting lots of cytokine right where the challenge infection is. Okay, so early Th1 immunity, this rapid immunity, that's what correlates with protection. So we can use adoptive transfer systems to formally demonstrate which of these activated T cells, these antigen-experienced T cells, are actually conferring protection, are actually able to home rapidly to a site of challenge and confer protection. So again, CD44 is this beautiful marker that distinguishes antigen-activated T lymphocytes from naive T lymphocytes. So you stain for all of the CD44 high cells, and then this CD62L, its expression is lost on the effector cells, so they're in the recirculating pool. CD62 ligand remains high on the central memory cells, so you can sort these two populations. In each case, they're CD44 high, but one is CD62L high and one is CD62L low. And then you sort those populations. Now you have them in a tube, and now you inject them intravenously into a recipient mouse. And then you infect those mice 1 day later, and you look for the the recruitment of those cells to the challenge site, and you look for protection. So the first thing you'll see, this is, so these are the recipient mice that have received the effector cells, the 44 positive CD62L cells, and been challenged, and then we're looking 5 days after challenge. There's a beautiful population of the effector cells that have been recruited to the challenge site and are secreting interferon gamma. So these are the right cells for protection. They're getting to the right place, and they're getting there pretty quickly. The memory, these central memory cells, the CD44 high, CD62L high, you don't see them in that challenge site for the first 5 days. So that's a problem. They're not going to be helpful in conferring protection if they're not even getting to the site. If you look at 12 days, now it's hard to find your effector population. But that's okay. They've already done what they needed. They got there early, and they hopefully did what they needed to do. Now you begin to see your central memory population. And we put in a proliferation dye, and because the proliferation dye has been diluted, it means the cells have been divided. So it tells us that the central memory cells first underwent a lot of division, and maybe that's why it took them a while to then actually differentiate to effectors. So they are good effectors. They're making interferon gamma. It just took them a long time to get back to where they need to be, okay? So then this is just looking now at protection. This is just looking at the numbers of parasites in the skin 21 days post-infection. The effector population conferred at least some reasonably good protection in the challenge site in the ear. Again, we didn't transfer a lot of cells, so maybe that's why the protection is not so strong as we'd like it to be. But the central memory cells didn't confer any protection at all. If we look in the draining nodes, the protection is better. So this tells us that central memory cells are not going to be a sufficient population of cells to confer protection against this kind of infection. But remember, it's central memory cells. These are the cells that everyone thought you needed, and that's what vaccines would generate. And those were the target populations. But for these kinds of vaccines that require T-cell mediated immunity, central memory cells are not a sufficient cell. It suggests that at the time of challenge, you need effector, you need cells in the effector pool. And then this experiment is just testing the lifespan of these effector cells. And now the effector cells are the key cells that work in this immunity. Let's just test the lifespan. We're essentially going to do the same experiment, sorting the effector and the central memory and the effector memory pools. And we're going to transfer them to a recipient mouse, and we're going to challenge on day 1. But we're also going to just wait, let the cells sit in that recipient mouse for a couple of weeks, and then challenge. And see if they'll still work if you delay the time of challenge. So remember, when we transfer them to the recipient mouse and let them sit there, there's no antigen there. This is a naive mouse. We're just transferring these activated T cells to a naive recipient. And they're sitting there without antigen until the time of challenge. So these are the different populations, and we're going to look in the ear just at the recruitment of cells. So this is the critical data set here, because we've already seen this. This is the number of cells recruited to the ear. The effector population, the CD44 positive, LY60 positive, if we challenge 1 day, those effector cells, we see them in the ear, okay? we're challenging 1 day after transfer, and then 3 days later we're looking at the tissue, and we find the effector population, and I showed you in the last slide, but the central memory cells, they don't get, I'm sorry, the central memory cells, they don't get there. The critical experiment here, if we just let the cells sit in that recipient mouse without antigen for 2 weeks and then challenge, you don't find them recruited to that challenge site anymore. So in that 2 weeks, in the absence of any antigen, they presumably just died because they're short-lived cells. And so they need to be constantly turned over. And the reason the healed mice are so immune is they have persisting infection, so they constantly renew this population of effector cells. But the vaccinated mice that have received a non-living vaccine, they can't maintain those cells. They can only maintain central memory cells, and they don't get there quickly enough. So in these challenge sites, again, it's these effector cells that confer pretty strong protection in these recipient mice. And the central memory cells that have been depleted of these effector cells, they don't work nearly as well. Okay. So the conclusions, chronic primary infection with Leishmania major maintains these high frequencies of effector cells that are short-lived in the absence of infection. So these cells, they reproduce the unique properties of protective response, rapid proliferation independent. These are cells that don't have to first proliferate to do anything. They produce a lot of gamma. They're already ready to produce gamma because they're effector cells. As soon as they encounter antigen in the tissues that they find it, they'll secrete the cytokine. And importantly, they're tissue homing. And these are the cells that are maintained in the healed animal and recruited rapidly to the skin in infected fly challenge. So protective immunity in the L major model of concomitant immunity is not mediated by memory cells. And this probably is what goes on in other chronic infections. So like TB is a chronic infection known in individuals with latent TB, and animal models have demonstrated this. If they get challenged with a new strain, they're fantastically immune, even though they haven't gotten rid of their primary infection yet. It's because they're generating these populations and maintaining these populations of effector cells that confer beautiful immunity to secondary challenge. So the protective immunity in individuals with healed infections is used as a rationale for vaccination, but this immunity may not be defined solely by memory. The failure of Leishmania vaccines against cutaneous leishmaniasis may be related to the fact that conventional vaccines cannot maintain a population of pre-existing terminally differentiated effector cells. So again, the key point is the effector cells have to already be present at the time of challenge. They can't be generated by this delayed response of central memory cells that takes a few days. They have to already be there. So vaccines may have to accommodate the requirement for infection or persisting antigen to establish and maintain the protective response. And that is the rationale for live attenuated vaccines. And there's tons of experimental work done. This is just a list of some of the live attenuated vaccines against leishmaniasis using physically attenuated, chemically attenuated, or genetically attenuated parasites. You know, nobody likes to use live vaccines because they're hard to deal with, they're hard to standardize, they're hard to deliver as compared with non-living recombinant protein vaccines that you just put in a vial. So nobody likes these things, but there's an argument that these are the only kinds of vaccines that will work against these kinds of diseases. I'll just make the point that this same vaccine that was used in these prophylactic trials like in Iran that did not work prophylactically. What if the problem with Leishmanization with L major is that the lesions are chronic and sometimes they're just really nasty, so you're just reluctant to give them to children. So what if you just mix the killed vaccine with the live vaccine? And so we've done that in mice and in monkeys. And so this is the primary lesion. If you mix it with just the heat-killed vaccine plus CpGs as adjuvant, you attenuate the pathology of this live vaccine, both in terms of pathology and the numbers of parasites. So we know this is Leishmanization. Once these heal, if they get re-challenged, it's beautiful immunity, and so that's what we're seeing here. This is the re-challenge of these mice here compared to the control. But importantly, when we've attenuated the vaccine lesion by just adding the killed vaccine to it, we didn't reduce the level of protection. So if a big concern of Leishmanization is just how nasty those lesions are, there's ways to genetically attenuate it, but there's a simple way to do it with a vaccine that's already been experienced in these trials in which thousands of people have already been given this vaccine and it's been found safe. So why not just attenuate the virulent strain by just mixing it? So that's an option. And then there's this issue of, will Leishmanization with Leishmania major protect against a visceral disease? So we had discussed that as an interesting possibility. So again, Leishmanization still is the only experience in people that confers any protection against any form of leishmaniasis, but it's only been used to demonstrate, to confer homologous protection against L major in those various places like Iran and Israel and Russia. So we addressed it experimentally. We immunized with Leishmania major in the foot pad, and then we challenged in the ear with a visceral strain, Leishmania infantum from Spain. If you put a lot of parasites in the ear, they will visceralize, and you get some infection in the liver and spleen. It's not progressive leishmaniasis. There's some pathology in liver and spleen, but the mouse is a terrible model for human disease. Mice really, no matter how many parasites you give them, they do not develop progressive disease, but they will develop infection. So we're just looking at protection against parasite burden in the viscera. There's a lot of data, but really the only relevant data is here. We're looking in the spleen and the liver 6 weeks after challenge with the visceral strain. So the mice that have been leishmanized in the foot pad with L major do have, in some cases, it looks like sterile protection in the viscera, in the spleen and the liver against challenge with the visceralizing strain. So there's cross protection. There's cross species protection conferred by the cutaneous L major strain. This wasn't so surprising just it was already known that they share a huge number of antigens, both pro-mastigotes and amastigotes. The Leishmanization with L major maintained a high frequency of Th1 cells that were retained in the blood vasculature of the liver and the spleen. So they were well poised to get into the tissue and localize into the parenchyma tissue after the challenge. So Leishmanization with L major may be a viable strategy for prevention of human VL. Okay, and I throw this up there because in ways I think it's similar to the first vaccine that was ever developed, which was, of course, using cowpox or variola to protect against smallpox. So cowpox was a pox virus of cows, produced mild disease in humans, but it cross-protected against this horrible and usually fatal disease of smallpox that in humans, right? So L major is primarily a disease of rodents. It produces, fairly mild disease in humans, and it cross-protects against anthraponotic, these human strains that produce a killing disease. So in my mind, there's similarities. In that context, you could say live L major, even if you don't genetically attenuate it, is still an attenuated strain the same way that cowpox is an attenuated in the context of what it does in humans, right? So the trick is getting programs, this sort of concept to be accepted, getting ministries of health to accept these kinds of ideas and to deliver another human pathogen. So in India, do you really want to put Leishmania major, which is a human pathogen, do you want to introduce it into these areas that are endemic for visceral leishmaniasis and, as we've discussed, actually have the right sandflies for L major transmission? But there's no L major there yet. So you're potentially introducing another human pathogen to that biotope, even though we're protecting against visceral disease. But there are absolutely ways to genetically attenuate L major so that it cannot be transmitted by sandfly bites. So we can knock out the LPG. So it will confer still immunity in people, but will not be able to be introduced into a natural transmission cycle. So why don't we stop there and take some questions. It'd be nice to hear your ideas about what I've just discussed and whether you're encouraged or discouraged about prospects for vaccines against any of forms of this disease.
Okay. Sir, we can use stationary phase, this L major strain for immunization purpose for VL patient, या procyclic या metacyclic, which stage?
Well, you want to produce a lesion.
Yeah.
So you need to use an infective stage. But your point is, maybe if you use procyclics, you'll still immunize without producing much pathology. I think there has to be a take. There has to be an infectious process. And procyclics, they won't give much of a take. It's similar to when you, none of you guys got smallpox vaccine, but I did, and so you were delivered live virus, vaccinia, and if it didn't take, if you didn't have a scar, they had to do it again, so it had to grow. But if your point is, why not just give procyclics and you'll minimize the pathology, and will it still protect? Actually, I think that's a perfectly good idea. there has to be a take. So there needs to be some infectious parasites, or it's not going to work. Yeah.
In previous slide, you use for immunization purpose, which type of mice you use, because in C57 they are resistant to parasites because Th1 response is very high. Then how we can say, suppose we use 10 to the power 4 parasites, that parasites generate memory and they kill the another pathogenic parasites like 210-6. Then how you can say?
So yeah, so we're immunizing B6 mice. We think B6 mice is the right model because, again, most people are resistant. They develop lesions that then heal, so they do develop acquired resistance. The point of the vaccine is to have the immunity much quicker so they don't develop a lesion at all. So that's why the B6 model is the right model. Even though they will self-heal, the point of the vaccine is so that they don't develop any lesion. Not to make them develop a healing lesion, but to prevent disease, prevent cutaneous leishmaniasis completely, right? So that's why a resistant mouse strain is the right mouse strain for cutaneous leishmaniasis, yeah.
Then we can say for they generate memory, yeah, in hyphen gamma do something. Suppose we collected T cells for immunized mice and we co-culture with infected macrophages because antigen is present in, then hyphen gamma kill the parasites. Then how we can say it is only because of the develop the memory, yeah, hyphen gamma do something.
I'm not sure what your question is. So right, these T cells, particularly effector cells, produce interferon. If you co-culture with infected macrophages, that's what they would do. They would, and even central memory cells would do that. Central memory cells, if you co-culture, they'll proliferate and they'll also begin to secrete interferon gamma in a culture dish. But in vivo, the process of them actually being able to get to where they need to get to secrete cytokine and activate infected cells for killing takes a few days. Whereas effector cells get there within just one or very, very fast. Because that's what they do. They're in the recirculating blood. They're looking for tissue damage, sites of inflammation, and so they're tissue homing cells. So they just get there very quickly. So you wouldn't appreciate those differences just in culture. I think both those activated T cell would activate those infected macrophages to kill.
Thank you.
Sir, earlier in previous slide, you have talking about early response and late response through CD40 and CD40 ligand mediated interaction, maybe generally through IF and gamma mediated, through IL-12 mediated protections. But in that slide, in last phase, parasites are still remaining, maybe 10 or 100 something like that. Then persistence of parasite is necessary to maintain sterile immunity or mediated protection. Persistence of antigen or pathogen is necessary to mediate.
Right. So it's actually not so clear how the persistence works. Is the persistence working by activating? There's always a turnover of naive T lymphocytes coming in from thymus that become activated by persistent antigen, or do the central memory cells? So I don't want to give the impression the central memory cells do not contribute. I'm just saying they're not sufficient, because the effector cells that are constantly renewed may in fact be generated from some central memory cells in lymphoid tissue that that they may not arise from new naive precursors, that the central memory pool in lymphoid tissue, because of the persistent antigen, that's what gives rise to these constantly renewed populations of effector cells. So that's not clear, but it's actually an important question.
In a full-blown patient who are infected with visceral leishmaniasis, so in those cases, can we get the T cells and then generate those, sort the...
I'm sorry, which patients?
Full-blown visceral leishmaniasis, where it is at a higher stage of L-Donovani. So in those cases, can we get the T cells and then culture them in vitro, let them differentiate into effector memories, and then reintroduce, so that the toxicity of the drug can be reduced and we can get a much better...
Yeah, so the last discussion this morning, we'll talk about visceral. It'll all be human immunology data and visceral patients. So yeah, excellent question. And we'll bring it up again when we talk about the VL patients. Yeah. So it was useful to talk about acquired resistance first in these cutaneous leishmaniasis models, because both in the mouse models and humans, there is very excellent acquired immunity to those infections. And so before we get to these more severe non-healing Leishmania infection models like visceral disease, what's the sort of resistance, acquired immunity we're looking for? Well, in patients that do control their infections, what sort of immunity does it, and why isn't that occurring in visceral disease? And how can we maybe make vaccines work better to allow them to do that? So that's why we talked about cutaneous leishmaniasis first. But it is a discouraging thing if we can't develop a vaccine against cutaneous leishmaniasis, where we're just trying to mimic the natural immunity that develops in those people. It's going to be a whole lot harder to develop a vaccine against an infection in which there's not good naturally acquired immunity. That's really going to be hard. There was a question at the back.
Hello. Hello. Am I audible? Yeah. So actually, I wanted to ask whether the CRISPR-Cas technology can be used in Leishmania in the sandfly embryos, and then we can. So the sandfly will develop immunity, and then we can release these, because the life cycle will be completed, because this technology is being used in malaria. So I'm not sure about Leishmania.
Right. So CRISPR-Cas9 has been used for editing sandflies and to knock out the IMD pathway in sandflies. But that is a very challenging problem, to generate these transgenic flies and then release them into natural biotopes and have them repopulate and displace a natural biotop. But experimentally, for sure, CRISPR-Cas9 editing works very well in sandfly embryos. Yeah.
Do you know any group who is working on such lines?
So the only group that has done CRISPR editing in flies is our group. So there's just not a lot of group that have sandflies.
You have any data on that?
So if you give me your email, I'll send you the paper, the manuscript that is not yet in press but soon to be. So, okay.
Thank you, sir.
Yeah. If you do the reverse, yeah, we didn't try that. I mean, that's not an approach you would ever use. You wouldn't infect people with Donamani to protect them against cutaneous. But experimentally, that's a, you know, that's sort of an interesting question. I presume these mice that don't develop much pathology from high-dose Donamani, in fact, do those infections in the liver and spleen, do they confer a good immunity against cutaneous leash from L major? I bet you they would, but I don't know actually. Donamani just doesn't provoke much of an immune response in mice. Doesn't grow well in mice, doesn't provoke a strong immune response in mice. It's not a mouse pathogen, it's a human pathogen. So the mouse is a difficult model, unfortunately, with Leishmania Donamani. So. Okay, shall we, one more question? I'm sorry? So in India, the langur monkeys, at CDRI, they were using langur monkeys for VL and to test vaccines. But it's very expensive. But for the visceral model, that's the only monkey model that I know of. Other monkeys have been used, spider monkeys,
Cannons are also...
What?
Cannons? Dogs?
Yes, so, well, yeah, so that's a good point. So dogs are a natural host of Leishmania infantum, not Leishmania donavani. But dogs are, because they get naturally exposed, dogs are a fantastic animal to test vaccines against natural infection. And since canine leishmaniasis is so prevalent in places like, you know, Spain and Greece, Italy, these high endemic sites for dog leishmaniasis, if you had a vaccine that you wanted to test against a proper natural exposure, dogs are a great model. And then you could argue, well, this vaccine worked in dogs against visceral leishmaniasis, maybe it would work in people. So dogs could be really an important tool for development of a human vaccine. Sadly, there's more money by drug companies devoted to developing vaccines against dog leishmaniasis, because these are pretty wealthy countries. Spain and Greece and France and Italy, they all have canine leishmaniasis. Pets die of this disease. So their owners would be willing to spend hundreds of dollars for a safe and effective vaccine. So the only drug companies that have ever put money into developing vaccines against leishmaniasis are veterinary companies. They couldn't care less about human disease. But that said, a lot can be learned from that experience in dogs, right? So, for challenge, yes. Yeah, that's a good question, because mosquitoes are used to test, infected mosquitoes are used to test malaria vaccines. And that's the only way they test malaria vaccines. They don't use needle. The difference is you can drug treat malaria. So these volunteers who get immunized and then challenged with infected mosquitoes, you know, they monitor their infection in the blood. And as soon as the infection gets a certain level in the blood, they treat with drugs that completely eradicate the infection. So we don't have drugs that completely cure or kill off leishmaniasis. So it's harder to convince you know, ethical review boards that live sandfly challenge should be used. But it would be it's the much better way to do it. Because right now the only way to test vaccines is is actual field intervention trials, which are incredibly expensive. Whereas if you could just challenge them in the laboratory with sandflies, you could do it so cheaply and just greatly reduce the numbers of people that are vaccinated to test the vaccine. But I think that's the main issue. You would have to convince institutional review boards that well, we don't think when they get infected they're ever going to get rid of these parasites, but we don't think that's a problem. But it may be a problem, so that's the problem. But that's a good question. Yeah. You can infect sandflies with other strains? Oh, with attenuated strains. Well, but how would you evaluate the protection if you can't evaluate with an attenuated strain? The protection has to be against a virulent strain. Okay, those were great questions. Why don't we take a break for how long? 15 minutes? Okay, start? Sure. All right. So the last subject for discussion is about human visceral leishmaniasis in India. So this is the discussion most relevant, of course, to the public health problem in this country. And we'll discuss work over the past many years done primarily in Professor Shyam Sundar's group at BHU, looking at immunity and immune regulation in human disease. These are just some of the folks. There have been so many people over the years involved in this research. I thought it'd be useful to, because I didn't show this before, just the history of leishmaniasis research in this country for historical purposes is so critical to just the fundamental discoveries regarding leishmaniasis generally. So no leishmania parasites had been observed or described until in 1903 in India. Major Charles Donovan and Major William Leishman, they independently discovered these parasitic bodies in the spleens of patients dying from an anomalous type of Indian fever, Kala-Azar, black fever. Again, these patients had a discoloration of their skin. So a long time ago. And so it was subsequently named Leishmania from Leishman and Donovani from Donovan by Ronald Ross, who was, of course, one of the most famous parasitologists in the world, who had discovered the transmission cycle of malaria. I went to see William Leishman's grave in London, in Highgate, London, and I was so shocked to see that it had been pushed over. So leishmaniacs around the world should be concerned, but we should get some effort together to go to the cemetery in London and resurrect William Leishman's tombstone. So the vector, the first demonstration that sandflies transmitted leishmania was also in India. The first natural infection of a sandfly, Phlebotomus argenipes, in North Bihar reported by Short in 1926. Its vector potential was proven by Swaminath in 1942, who transmitted the parasite to man by the bite of experimentally infected Phlebotomus argenipes. Okay, so this is the disease in India. We've gone through clinically what this disease looks like, and we've also made the point that in India, as in the majority of endemic zones for visceral leishmaniasis, the majority of exposures to this parasite are asymptomatic. This is the ratio of incident asymptomatic infection to symptomatic clinical disease. So in India, it's 8 or 9 or 10 to 1. So many more people somehow control the infection after transmission by bite than go on to develop disease. So a lot of work. So though these asymptomatic individuals, I guess, are considered immune. They have some sort of either natural immunity or naturally acquired resistance. They somehow controlled these infections before these parasites developed full-blown disease. So this just summarizes a whole lot of work done in places in India and in Sudan and in Brazil, looking at asymptomatic carriers and the nature of their immunity. So just a lot of cells and cytokine here, but essentially it looks like the Th1 immunity we were looking at in cutaneous leishmaniasis. So, and so CD4 cells making IL-12, acting upon differentiating to Th1 cells and secreting interferon gamma, and TNF is always an important cytokine associated with resistance in the visceral, in these asymptomatics, and then upregulated NO synthase in both macrophages and monocytes in these individuals. In addition, another pathway has been described by 2 or 3 investigators, mainly in Sudan, that involves Th17 cells, not Th1 cells. So some cytokines, it's thought IL-6 and IL-23, act on precursors to differentiate to Th17 cells, which secrete IL-17, which somehow promotes resistance in these infected cells, whether it via upregulating IL-1 beta is not known. This is a very important study done in Sudan, in which it was a 6-year longitudinal study on 1,600 subjects, in which they identified incident exposures. So using immunologic assays, I think, so these were healthy individuals who they, from immunologic assays, seroconversions, I think they defined as having been infected, but were still healthy. Then they followed over 6 years and determined who went on to develop Kala-Azar and who remained resistant. But at the time of these incident exposures, they took their lymphocytes, they stimulated with antigen-collected cytokines and defined the different cytokines in those supernatants and froze them down. And they found that the 2 cytokines that associated most with, over here, with resistance, individuals who did not go on to develop Kala-Azar, they made high levels of IL-17 and IL-22. And these cytokines were absent in the supernatants of healthy individuals who then went on to develop disease. So somehow these Th17 cells, which have been thought mainly to function in acquired resistance to extracellular bacteria and fungi, may play some role in immunity to these Leishmanian infections. So that was a very interesting study in Sudan. And then I wanted to call your attention to the most important human genetic study looking at risk factors, why some people develop disease and others don't. So again, this was the work done, directed by Jenny Blackwell in collaboration with the folks in Shyam Sundar's program. It took many years. And this was looking across the entire human genome, looking for genetic risk factors for visceral leishmaniasis. So these are whole genome scans. These are the different chromosomes. In the Indian discovery data, it was over 500,000 SNPs that were looked at, and 989 cases of VL. So that's an enormous cohort, and 1,089 controls from the same endemic region. And then they repeated it in a Brazilian discovery data set, again over 500,000 SNPs, single nucleotide polymorphism, 357 cases in 308 Brazilian families. This is the meta-analysis of the 2. So the SNPs in red show regions with replicated association to visceral leishmaniasis susceptibility. So this is truly remarkable across these huge geographic spans, and really different parasites. One is Leishmania infantum, one is Donovani. They both produce visceral disease, but in these two very different outbred human populations, the same genetic locus was associated with the risk of developing disease, and these were polymorphisms in the HLR-DRB1 locus. So this is an immune response locus, and it really is quite an impressive and strong association that suggests that there is an immune basis of a genetically encoded immune basis to susceptibility, that at least in part, the genetics is a risk factor for who develops disease and who doesn't. It does suggest that there may be a hierarchy in the antigens. The fact that this is an HLA region essentially associated with presenting antigen to CD4 cells. So it suggests that there is a restricted set of Leishmania peptides that are loaded onto this HLA molecule that if it's when that occurs, these individuals don't develop a protective response, but instead develop susceptibility. So that needs to be followed up in terms of just what mechanistically the nature of this HLA locus is doing to confer this risk. So that's the other really important study that has been done over the past half a dozen years or so. So now getting to the work done up at BHU, looking at the immunologic defects underlying the pathogenesis of human VL. So looking in cases with active disease, usually pre-treatment, and comparing with post-treatment. So human disease in India and elsewhere for years had been associated with antigen-specific unresponsiveness of PBMCs for gamma secretion or T-cell proliferation. That was thought to be a hallmark of visceral disease. So if you took their peripheral blood cells, you have to keep in mind that when you do human immunology studies, you can't be getting a lot or any lesional tissue. The analyses are confined to peripheral blood. So you may be missing a lot, but that's what you have to look at. And what had been observed for years is peripheral blood cells from active cases. If you stimulated with antigen, you just saw no response. There was no interferon gamma response. If you stimulated with a mitogen, then they responded well enough. But they had lost an antigen-specific response. So that was a hallmark of disease. So work done up at BHU starting, I don't know, 7 or 8 years ago. They adapted this quantiferon TB assay, which had been used for the diagnosis of latent TB infection, which was just a whole blood assay. You don't purify the peripheral blood leukocytes. You just take whole blood and put it in a tube, and they stimulated with antigen and then took and put it in culture for 1 or 2, 3 days, and then took the culture supernatants of the whole blood and just did ELISAs for cytokines for interferon gamma. Simple assay in response to this TB antigen. So it was adapted by the group at BHU. This quantiferon assay was adapted using Leishmania antigen instead of TB antigen. So the whole blood assay. And the point was to detect subclinical infections or asymptomatic infections with Donomani. So when they stimulated whole blood from non-endemic human controls, non-endemic healthy controls, they made no interferon gamma. But when they took, well, let's look at the positive controls. These are cured VL patients that were thought to be, that had visceral disease, immune response to Leishmania, Donomani. The majority of them made a pretty good response, interferon gamma response. But amongst the endemic healthy controls from the endemic region, about a third of them responded. So these were considered as exposed and immune individuals. So the only other way they had identified asymptomatics before was seroconversion. But in fact, this is a better way to identify exposed and immune individuals. The fact that they're making interferon gamma and they're healthy suggests that they really have been infected, but made the right kind of response to keep them healthy, to control the infection. So that assay worked pretty well for identifying these subclinical infections. What was surprising was when they then looked at active VL cases before treatment, they had full-blown disease, most of them made really high levels of interferon gamma. So that was completely different than what had been widely accepted, this antigen-specific unresponsiveness using peripheral blood cells, using this whole blood assay in which you don't manipulate the cells, you don't spin them through a Ficol gradient, and then take these peripheral blood leukocytes and put them in a tube. Just the whole blood, you don't screw around with it, just throw in antigen, and they secreted interferon gamma. So they had disease, but in fact, they still did have lymphocytes that made interferon gamma. So that was a surprise. And then they found that it was mainly, if they took the whole blood and they depleted the CD4s, the levels of interferon gamma in these whole blood assays went down. If they depleted CD8s, nothing happened. So it suggested the response was mainly produced by CD4 cells. They could block the response with antibodies against HLA-DR. So it was an antigen, a class 2 restricted T cell response. This is just by flow cytometry intercytokine staining in cells stimulated with SLA, whole blood cells. It was CD4 cells, and you could see some staining for interferon gamma. And there was a nice association between the percent interferon gamma positive T cells in this staining assay and the levels of cytokine released in culture. Okay, so even people with active progressive Kala-Azar, they actually have a residual Th1 response. I know there was some interest in CD8 cells in Leishmania infections, including, I already showed this data that in these whole blood cultures, if you depleted CD8 cells, there was no change suggesting CD8s didn't do it. What's important, cured VL cases reconstituted a CD8 gamma response. So the percent of CD8 cells making gamma in response to Leishmania antigen goes up. And if you remove CD8s, the amount of gamma and all of the peripheral blood cells goes down. So CD8 cells can make gamma, but they're not doing it during active disease. And the group at BHU had a nice study to show that the CD8 cells show markers of energy and exhaustion in human VL. So CTLA-4 and PD-1 are these markers of energy or clonal exhaustion. These have been the targets of these checkpoint inhibitors that reconstitute immune responses in cancer patients, and for which a Nobel Prize was given just this year. So T cell exhaustion is usually seen at the level of CD8 cells, and in patients with tumors, their CD8 cells can be potentiated by blocking these markers of energy and exhaustion. So there's elevated CTLA-4 levels in the PBMCs and CD8 cells in these patients, pre as composed to post and compared to endemic controls. that's the percent. What is that? So then, in terms of PD-1, especially if they looked in splenic aspirate cells, the splenic biopsy tissue, the level of which is the lesional tissue in the spleen, the frequency of cells expressing the PD-1 marker is quite high. So CD8 T cells may be driven to energy and exhaustion in human VL, which may affect their ability to contribute to the protective response. So that was a nice finding. I guess it does suggest that maybe these checkpoint inhibitors would be therapeutic in human VL. And since they have now been given to tens of thousands of cancer patients, the drug is so well understood clinically. So let's go back to interferon gamma. So the thought was, well, we looked in peripheral blood, these whole blood assays, these patients with active disease made a lot of interferon gamma, but maybe it's not doing anything because if you look in the actual infected tissue, like the spleen, you won't find it there. But this is just looking at message because it's hard to do cellular assays in the tiny amount of splenic biopsy tissue that you get. So this is doing qPCR on mRNA for gamma. It was elevated in pre-treatment as compared to post-treatment. So in fact, they did, and it was elevated in patients with active disease compared to, well, it was at least as high as these healthy individuals from splenic biopsies from healthy individuals. So even in lesional tissue like the spleen, there seemed to be an ongoing interferon gamma Th1 response. So why isn't it working? So we're going to start to discuss a lot of data that has been generated at BHU regarding IL-10. So in addition to gamma message being elevated in splenic aspirate cells, IL-10 message was also elevated, comparing in the same individual day 0 to day 21 post-treatment, and comparing as a cohort these active cases to post-treatment and healthy individuals. Okay, so they have IL-10 message. The role of IL-10 in the pathogenesis of visceral leishmaniasis is quite well accepted now. IL-10 is an immunosuppressive cytokine. It was described in the early 1990s. And it's produced by many cell types, including T cells, B cells, macrophages, dendritic cells. It suppresses multiple antigen presentation and microbicidal functions of dendritic cells and macrophages. In animal models, in vivo neutralization of IL-10 enhances immunity and improves treatment regimens for VL. And elevated levels of IL-10 has been a consistent finding in experimental clinical studies of VL. So it really is absolutely characteristic of disease. This is just looking at plasma levels of multiple cytokines in active cases compared to endemic normals. And just the plasma levels of IL-10 are very characteristic of active VL cases. So they're secreting, putting out a lot of IL-10. And if we go back to the whole blood assays, so remember before, the whole blood assay in terms of gamma secretion didn't distinguish between active patients and drug-cured patients who are considered immune and asymptomatics who are responsive and considered immune. So their ability to secrete gamma was not diagnostic of active disease, but IL-10 secretion was. So antigen-specific IL-10 responses was not seen in these endemic normals, or when these patients are cured, their ability to produce IL-10 in response to antigen goes down. So active disease is associated with cells producing a lot of interferon gamma, and most of the cells, not interferon gamma, IL-10, most of the cells secreting IL-10 are T cells. this is doing some interesting assays with splenic aspirate cells, which they no longer do routinely now, but used to routinely do to confirm the diagnosis of VL before starting drug treatment. So fine needle splenic aspirates taken, put on a slide to stain for infected cells, and then the residual amount of aspirate, just a small volume, but containing, you know, a packed amount of leukocytes, usually over 1 million cells, could then be analyzed. And in this case, they just took ex vivo explants of these residual splenic aspirate cells containing infected macrophages and T cells and B cells, everything in these spleens that they happen to capture in the aspirates, and put them in culture for 3 days with and without antibodies to IL-10, antibodies to human IL-10. And then after 3 days of culture, they quantified the amount of parasites, viable parasites, that are remaining in those cultures. So this was done on over 60 individuals. So the first thing you'll appreciate is in terms of the numbers of tissue A mastigotes in these cultures from the splenic biopsies, huge range, and that's because these patients come in at different stages of disease. The needle aspirate is capturing different amounts of cells or different affected parts of the spleen. But in each case, you know, the readouts are going to be paired with and without anti-IL-10 antibody. So already it was remarkable that including anti-IL-10 antibody in these cultures of splenic aspirate cells potentiated the killing, the clearance of Leishmania donnelmanni from almost half of the cultures. So this is as close as, you know, you sort of get to actually treating people with a drug, with an anti-IL-10 drug, and showing an anti-parasitic effect. This is taking lesional tissue, not manipulating it at all, just blocking one cytokine and allowing the cells that are sitting there to function when you block the activity of that one IL-10 cytokine. And if you then measured the levels of interferon gamma and TNF that are secreted in these splenic aspirate cultures, it was almost always higher when you included anti-IL-10 antibodies in these cultures. So this really strongly argued that anti-IL-10 would be therapeutic in these patients. Now keep in mind, these patients are making IL-10, if you remember the first lecture I gave, IL-10 is an immune regulatory cytokine that when it was blocked, or when IL-10 genetically deficient mice were infected with toxoplasma, they died by a cytokine storm. So these individuals, they're making IL-10 for a reason. So now we know, in fact, there's a lot of pro-inflammatory cytokines in this lesional tissue in the liver and spleen, and the IL-10's trying to dampen, you know, the immune pathology that can be mediated by these responses. But at the same time, it's suppressing the ability of those immune cytokines like interferon gamma and TNF to kill. So it's this delicate balance between immune-mediated pathologies and anti-parasite activities. But it does suggest that by inhibiting IL-10, the balance between effector regulatory cytokines can be shifted to promote more effective killing of the parasite by activated macrophages in the spleen. So anti-IL-10 antibodies or IL-10 receptor blockade should be considered as an immune-based therapy alone or in conjunction with drugs to reduce the dose or the length of treatment. Already, I think they're convinced that the drugs that are used if there's no residual response, like in HIV co-infection, the drugs don't work very well. So the drugs that are used are already require some intact immune response. So with anti-IL-10, you could make that level of immunity even higher, and the drugs would be more effective. So this would be, you know, a therapeutic, a host-directed therapy, a therapeutic vaccine, which really would have a very strong chance of working quite well. So I want to end this discussion with the recent work that's being done up there on quantifying infection reservoirs in human VL by xenodiagnosis. So this is work funded in part by the Gates Foundation, and who found it critical as part of the Kala-Azar elimination effort in India, Nepal, and Bangladesh that that they needed to understand how the transmission cycle is maintained. So just considering the possible infection reservoirs, clearly patients with active disease, patients with PKDL, cured cases. So do cured cases still harbor parasites? Can they transmit? And most importantly, the question was subclinical cases, who represent the preponderance of clinical exposures. Do they help to maintain transmission cycles? So they were desperate to answer that question. And then also this question of non-human reservoirs. There are still those who think that there are non-human reservoirs who contribute to the maintenance of transmission cycle, particularly between these sort of epidemic periods of when the disease is cycling down and then back up. Still, there's no evidence at all yet for non-human reservoirs. So how can xenodiagnosis aid elimination of VL? It can understand the transmission dynamics of Donnellmani infection, help resolve the long-standing debate on the role of asymptomatic infection in disease transmission. It can measure treatment efficacy. So after drug cure, do these individuals still harbor parasites that can be transmitted, and resolve the role of PKDL in disease transmission. So xenodiagnosis, for those of you who may not know, it's the detection of a parasite by feeding supposedly infected material, like blood, to a suitable intermediate host, like a sandfly, and later examining the intermediate host for the parasite. So xenodiagnosis was originally used to diagnose in South America patients with Chagas disease, the American trypanosomes. They didn't have any sensitive way of detecting them, so it's transmitted by these reduvid bugs. So they would feed reduvid bugs on these individuals, and those reduvid bugs would propagate in the vector, and they could then diagnose Chagas disease. There's direct xenodiagnosis. Insect feeds directly on the host, and indirect. You bleed the host and then feed the insects on the blood from that host. The xenodiagnosis studies we're doing here is all direct feeding of human volunteers exposed to sandfly bites. So to expose people to the bites of sandflies, you can't use wild-caught flies because they may harbor parasites. You have to establish a laboratory colony of flies and cycle it through many generations and validate that it is parasite-free and free of certain viruses that phlebotomines can also carry. And establish that those colonized flies are safe to use for exposing humans to. So they establish colonies by going out to the field and collecting Phlebotomus argentipes and not these other flies which they found. And these are the sorts of sites they use, light traps. They would put them out at night and go the next morning and collect the flies. The insectary was built at the Kalazar Medical Research Center in Muzaffarpur. So a beautiful insectary was built. These rooms look bigger than they are. They're actually quite small, but more than adequate. And they succeeded in establishing really an incredibly healthy colony of Phlebotomus argentipes. They produce well over 1,000 adult females each week that can be used to feed on people for these xenodiagnostic studies. So they would collect the flies, put them in these containers, which are then strapped on the arm, and then the flies are housed in an incubator for 2 or 3 days, and then dissected and microscopically observed for the presence of parasites, or PCR'd for presence of parasites. So xenodiagnosis has been done in VL, but not more than really just a handful of cases. So these are studies involving hundreds of people, so it's an extraordinary study across all the exposure groups, including active cases, cured cases, asymptomatics, and PKDL. They did a lot of work to optimize the timing and site of sandflies feeding success. There was some concern that when they were feeding during the day, the flies might not bite so efficiently. So they compared day and night and different sites on the leg and the arm. At the end, they found it was as efficient to feed during the daytime, certainly more convenient for the patients and the volunteers and the investigators than at night. Of the leg or the arm, it didn't make so much difference. So they're feeding these individuals with 30 flies on the arm and 30 flies on the leg, 60 flies total. That doesn't mean they all take a bite, doesn't mean they all survive the 2 to 3 days for dissection. So this is number of active cases. This is just the data from that. So already this is xenodiagnosis on active disease. They had an interesting finding that this is the proportion of flies infected with the parasite, percentage of flies infected in cases with a splenic score of 1+, 2+, 3+, or 4+. So 1+ is early stage of disease, and 4+ is much later stage. So not surprisingly, the more severe the disease status, the higher percentage of flies take up, get infected. But what was particularly interesting, in the early stage of disease, these individuals are barely infectious to flies at all. So these are the relative odds ratios comparing to the 1+ group. The odds ratios of these other groups. So these 1+s barely. I think there was only one individual that was scored 1+ that transmitted to flies, whereas there was very efficient transmission in the 4+ group, a late stage infection. So that's probably not so surprising, but still, these individuals, they have full-blown Kalazar. It's important to note that the earlier the diagnosis is, the earlier the treatment, and you'll remove them from the pool of individuals that can transmit to flies. And this is the most important group because this was really the rationale for the whole study, asymptomatics. They've done 156 so far, 86 males, 70 females. This is the sandfly feeding response on the arm and the leg. So, you know, an average of between 20 or so flies fed on each site. So usually there was in total about 30 to 40 flies that would be dissected. And none of the asymptomatic subjects has been able to infect flies. I think we were probably surprised at that, but that's an encouraging result, that they probably do not contribute to maintaining the transmission cycle. So you could really focus your attention in terms of maintaining the vector control effort on when an new active case appears, you do your insecticide spraying around that house. And you don't have to worry about all of these asymptomatics that are out there. Okay, so how about PKDL? Well, they started just a couple of subjects, both of them transmitted to flies, whether the flies were exposed to the lesions or to non-affected areas of the skin. So the lower arm as opposed to the nodular lesion didn't seem to make so much difference, so that's interesting. Those were just 2 subjects, but in fact they've done 26 more now, and I only got this data a few days ago, so I haven't had time to put it together. These arrows are these are 26 PKDL, some nodular, some macular. 11 of 26 were positive to flies, so these were the ones. They're transmitted to at least 1 fly. And in this case, the nodular lesions were more efficiently transmitting to flies than the macular lesions. Okay? So it is interesting that not all the PKDL patients transmitted, but certainly a high percentage of them did. And there's more PKDL out there, and so there's no question that they are contributing to the transmission cycle. So sandfly infectivity correlates with severity of disease. 42% of PKDL patients were infected to flies. None of the asymptomatics, none of the cured VL subjects, and I didn't mention this, when these PKDL subjects were cured in the data they just sent me, not a single one transmitted to flies. So the treatment even eliminated transmissibility in the PKDL patients. Okay. So there's a huge group involved in all of that beautiful work. So I think we'll stop there and take some questions.
So I wanted to ask, in the experiment where PBMCs were stimulated with Leishmanial antigens, so what they have used, the complete protein extract or specific proteins or some peptides?
Yeah, it's just a very crude antigen prep. Just whole soluble antigen, freeze-thawed promasticates that are then spun and you take the supernate. So it's a very crude prep.
And secondly, you have mentioned they have checked CD4 positive specific interferon gamma secretion by flow cytometry. So is it also be done for the IL-10 secretion, means cell-specific IL-10 secretion, or from the overall PBMCs?
Yeah, that's a great question. The data was up there. The data is not clear. We know, at least at transcriptional level, when we pull the CD4 cells out of the spleen, that they have upregulated IL-10. Now that's not protein, but in terms of message, that when we enrich CD4 cells from the VL spleen, that's where we see a lot of IL-10. In the peripheral blood cells, when we stimulated with antigen, it wasn't clear that the CD4 cells, when we knocked out the CD4 cells, we didn't affect the IL-10 response. Yeah, which was surprising. I'm still convinced CD4 cells are, look, a lot of cells do make IL-10, but we know CD4 cells do contribute to that response in an antigen-specific way, whereas macrophages make 10, and that wouldn't be antigen-specific, right?
One last question. In the case of CD8 cells, where they have shown the energetic surface molecules like CTLA4 and PD1 and all, in that case, it has been checked whether they are secreting some granzyme or they are just expressing these markers. Means when you are stimulating them with the antigen, you are actually not activating the CD8 cells. You are mainly activating the CD4s.
Yeah, that's a great point. So that's absolutely what they checked for granzyme secretion, which they did. So that's in that paper. So they looked at granzyme. Yeah, very good. Yeah. Any more questions? If not, I think we're having a photograph, right? Okay.
So you just spoke about the IL-10 therapy, right? So is there any recent reports about those things?
Is there any
recent reports about IL-10 therapy?
Yeah, we and others have tried to get drug companies interested in producing anti-IL-10 for this. So far that hasn't been possible. I think Merck has an anti-IL-10 drug that they're trying in some clinical setting, I think tuberculosis. So I do think at some point it will be possible. Yeah, there aren't many companies that have made anti-IL-10. But I think this is just the perfect clinical setting. I think IL-10 is upregulated in virtually any chronic infection. So a clinical demonstration that targeting IL-10 or receptor blockade can help improve the control of the pathogen in these chronic infections would be so important, so interesting, just as to proof of concept that this host-directed therapy is a way to potentiate an antimicrobial response. So, but I think this clinical setting of visceral leishmaniasis is a perfect setting to do that. TB is also an interesting setting to do that. And hopefully it will be done. Okay, shall we retire? Are we having photograph now?
On the entrance of the department.